Thanks for tuning in. I'll be discussing temperature-dependent interactions between parasites and gut bacterial symbionts of bumblebees. My basic question in this project was how temperature influences infection by affecting interactions between parasites, hosts, and the core gut microbiota. Temperature and symbiotic microbes are two factors known to affect resistance to infection. A common response to infection in humans and other animals is fever, an elevation of body temperature. These high temperatures can directly inhibit parasite growth, potentiate host immunity, and reduce infections in mammals, reptiles, insects, and even plants. Symbiotic gut microbiota can also improve resistance to infection by acting as physical and chemical barriers to parasite establishment. If temperature and microbiota can each affect infection on their own, how does temperature affect the balance between symbiotic and parasitic microbes? I broke this problem into three main questions. How do parasites and symbionts differ in thermotolerance? What are the effects of temperature on parasite-symbiont interactions? And how does temperature affect infection and gut microbiota in live bumblebee hosts? I first used cell cultures to characterize the temperature dependence of parasite and symbiont growth rates and antagonistic interactions. I then tested the temperature dependence of infection in live hosts. I'll be focusing on interactions between parasites and symbionts of bumblebees, which are important pollinators of crops and wildflowers, especially under cold conditions in which smaller bees are unable to fly. However, some species of bumblebees are threatened or in decline in the Americas, Europe, and Asia. Infectious diseases may be contributing to these declines. Two factors that could support resistance of bees to infection are their thermoregulatory abilities and core gut microbiota. Bumblebees have remarkable thermoregulatory abilities for insects of their size. They are facultatively endothermic during flight and incubation, able to maintain body temperatures that are astonishing 30 degrees above ambient temperature. This enables them to fly even at temperatures that are near freezing. Both bumble and honeybees harbor a core gut microbiota that is consistently dominated by bacteria from just 4 genera. These bacteria are acquired via social interactions in the colony and can enhance resistance to gut infections. I focused on the parasite Crithidia bombi, a trypanosomatid that infects the hindgut of bumblebees. This parasite is both prevalent and widespread in multiple bumblebee species on at least 3 continents. It has especially deleterious effects on queens that attempt to found new colonies in spring. However, the bumblebee gut microbiota have been shown to increase resistance to infection. Crithidia bombi and other trypanosomatid parasites of bees have recommended culturing temperatures of 25 to 27 degrees. Recommended culturing temperatures for all 4 genera of core gut symbionts are a full 10 degrees higher. This suggests that the core symbionts have higher thermotolerance than do the parasites, such that high or fever-like temperatures could eliminate parasites without harming beneficial gut bacteria. I examined interactions between Crithidia bombi and the symbiont, Lactobacillus bombicola. This is a dominant member of the bacterial community in the bumblebee hindgut where Crithidia infection establishes. To better understand how temperature affects interactions between symbionts and parasites, I use cell cultures to address 3 main questions. First, do thermal optima differ? For this, I measured changes in growth rate across a 25-degree temperature range. Second, is there evidence for antagonistic interactions? Do chemicals produced by symbionts inhibit growth of parasites? Third, if there are inhibitory effects, how do these differ across temperatures? And how do symbionts alter the optimal temperature range and thermal niche of parasites? To do this, I compared parasite growth across a range of temperatures with and without symbionts. I first measured growth rates of parasites and symbionts across a 25-degree temperature range. Here's temperature on the x-axis and growth rate on the y-axis. I found that the core symbiont, Lactobacillus bombicola, had higher optimal temperatures than did the parasite Crithidia. All 4 strains of parasites grew well at 27 degrees, growth rate peaked at 32 degrees, then declined to near zero at 42 degrees. In contrast, growth rates of the core symbiont, Lactobacillus bombicola, tripled as temperatures increased from 27 to 37 degrees, with strong growth all the way up through 42 degrees. Now, what temperatures might parasites encounter in their hosts? Remember, bumblebees are facultatively endothermic. Queen bumblebees generate heat in the nest to incubate eggs and larvae. Queens that have initiated nest building keep their body temperatures above 37 degrees. And queens can maintain these high body temperatures day and night. How about worker bees? Like queens, they incubate eggs and larvae in the nests and generally keep their body temperatures well above ambient, between 27 and 33 degrees. So now, let's return to those thermal performance curves to get a better idea of their physiological relevance. Incubating queens maintain temperatures that are higher than what's optimal for parasites, but ideal for the core symbiont, Lactobacillus. The temperatures of workers fall within the optimal range for parasites, but the difference between the low and high ends of their temperature range would make a big difference for the relative growth rates of parasites, where growth is relatively stable throughout that range, and symbionts, which grow about twice as fast at the upper end of that range. To summarize, the symbiont Lactobacillus bombicola performed best at temperatures that were higher than those of peak performance for the parasite, and had a comparative advantage at the upper end of the temperature range found in hosts. I then tested whether chemical exudates in the medium of the symbiont would inhibit parasite growth. The symbiont strongly and almost completely inhibited parasite growth. When parasites were incubated with spent medium, in which the core symbiont Lactobacillus had been grown, growth was completely inhibited. And the inhibitory effect was due to acid production and not nutrient depletion. When fresh medium was acidified, growth was likewise inhibited. Conversely, when spent medium was neutralized, the inhibitory effect disappeared. I found that the parasite's growth rate, shown on the y-axis, was inhibited by acidity levels within the range previously found in bee guts. With strong parasite growth at the pH found in germ-free bee guts, but more than 50% inhibition of all strains at the pH of symbiont colonized guts. So these experiments implicate gut acidification as a mechanism by which gut bacteria could protect against trypanosomatid infection in bumblebees. Moreover, parasite strains, shown on this axis, varied in their tolerance to acidity, shown on this axis, suggesting that gut microbiota could exert directional selection for pH tolerance in parasite populations. To summarize, the symbiont Lactobacillus bombicola inhibited the parasite Crithidia bombi via production of acids. Inhibition occurred within the pH range previously found in honeybee guts, suggesting gut acidification as a mechanism by which gut bacteria reduce infection. And parasite strains varied in their tolerance to acidity, suggesting that gut bacteria could select for pH tolerance in parasite populations. Do symbionts alter the thermal niche and optimal temperature range of parasites? To answer this question, I compared parasite growth rates across a range of temperatures in the presence versus the absence of symbionts. In the absence of the symbiont, Lactobacillus bombicola, parasite growth rate was highest at 32 degrees. However, presence of the symbiont at either of two initial concentrations slowed growth overall and lowered the optimal temperature for parasites to 27 degrees. The presence of the symbiont also had dramatic but temperature-dependent effects on parasite cell motility. Whereas monocultured parasites maintained their motility up to 37 degrees, there was virtually no motility of parasites above 27 degrees when they were co-cultured with symbionts. Symbionts grew faster and produced acids faster at higher temperatures, and the faster the growth of the symbionts, the slower the growth of the parasites. Returning to the original questions. First, thermal optima were higher in symbionts than in parasites. Second, the parasite was inhibited by symbiont-derived acids. and inhibition occurred within the pH range found in symbiont colonized but not germ-free bee guts. This suggests gut acidification as a mechanism by which gut bacteria reduce infection. Third, the different thermal optima of the parasite and the symbiont led to temperature-dependent outcomes of competition, with competition from the symbiont appearing to constrain the thermal niche of the parasite to lower temperatures. Having demonstrated temperature-dependent inhibition of parasites by symbionts in cell cultures, I then tested whether these ideas would apply to infection of live bees. Based on my cell culture experiments, I hypothesized that higher temperature would favor poor gut bacteria over parasites due to temperature-driven increases in symbiont-mediated inhibition and above 32 degrees direct high-temperature inhibition of parasite growth. Results in bees were consistent with what I had observed in cell cultures. Relative to the coolest temperatures, the highest incubation temperature resulted in an 80% reduction in infection intensity and the highest proportion of bees with no detectable parasites after 7 days. High temperatures appeared to protect bees against opportunistic infections by bacteria as well. Bees incubated at 37 degrees were half as likely to harbor bacteria from the Enterobacteriaceae, which includes opportunistic pathogens of bees and humans. One unexpected finding was that the parasite inoculation also reduced the prevalence of Enterobacteriaceae, suggesting that parasites could activate the immune system against opportunistic bacterial pathogens. To summarize the live bee experiments, high temperatures reduced infection intensity, consistent with predictions from cell culture experiments. In addition, high temperatures as well as parasite inoculation were protective against Enterobacteriaceae, a family of bacteria that includes opportunistic pathogens, suggesting that thermoregulation of bumblebees could improve resistance to infection, possibly by enhancing the protective effects of core gut symbionts. To conclude, these experiments show how temperature can potentiate the protective effects of gut bacteria against parasites. Core symbionts had greater tolerance of high temperatures than did parasites, and inhibited parasites via production of acids, with greater growth rates, production of acids, and inhibitory effects at high temperatures. As a result, symbionts constrain the thermal niche of parasites to cooler temperatures. The effects of fever on infection are typically considered in terms of host immune function and parasite infectivity. However, these findings suggest a novel microbiome-mediated mechanism by which fever could enhance resistance to infection, by augmenting the antiparasitic effects of non-pathogenic gut microbiota. I thank my colleagues, my families, and sponsors who have helped me over the years, the ESA for organizing this conference, and you for your audience. Please see the notes and the last slide for links to relevant articles, and leave a comment in the chat.